Your body constructs a perfect copy of its entire instruction manual over 1 trillion times every single day. This monumental task, vital for life, unfolds through an intricate dance at countless microscopic forks. At the heart of every living thing lies DNA, the blueprint that defines who you are. Every second, countless cells within your body divide, requiring a flawless duplicate of this genetic information. This crucial process, known as DNA replication, ensures continuity of life from one generation to the next. Without precise copying, genetic information would degrade rapidly, leading to cellular chaos and dysfunction. The central stage for this molecular drama is a structure aptly named the DNA replication fork. Imagine this fork as a bustling nanoscale factory, constantly churning out identical copies of genetic code. It's a fundamental biological process, ensuring growth, repair, and the very continuation of species. Understanding these forks reveals the incredible engineering underlying all biological existence. Prepare to witness the unseen architects of life, tirelessly working to replicate our genetic destiny. The first step involves unwinding the tightly coiled DNA double helix, like unzipping a microscopic zipper, creating the distinctive Y-shaped replication fork structure, forcefully breaking the hydrogen bonds holding strands together. This powerful molecular machine moves rapidly, creating the distinctive Y-shaped replication fork structure. Once separated, the two DNA strands must remain apart, preventing them from snapping back together. Single-strand binding proteins swiftly attach to these exposed strands, stabilizing them for the copying process. But simply separating the strands isn't enough. The actual construction of new DNA must now commence. An enzyme named primase then arrives, laying down a short segment of RNA called a primer. This primer provides a crucial starting point, a small handle for the main building enzyme to grasp. Enter DNA polymerase Roman III, the master builder, ready to synthesize new complementary DNA strands. It reads the exposed template strand and adds matching nucleotides, extending the new DNA segment with precision. One strand, known as the leading strand, is synthesized continuously and smoothly in a single direction. This effortless progression occurs because its direction aligns perfectly with the unwinding of the fork. However, the other strand, the lagging strand, presents a much more complex and intriguing challenge. DNA polymerase can only add nucleotides in one specific chemical direction, a fundamental constraint. Consequently, the lagging strand must be copied in short, discontinuous segments, working backward from the fork. These fragmented pieces of newly synthesized DNA are famously known as Okazaki fragments. Each individual Okazaki fragment requires its very own RNA primer to initiate its synthesis. So, primase repeatedly places new primers along the lagging strand as the fork expands. DNA polymerase then extends each of these primers, creating multiple small DNA sections. These numerous gaps and RNA pieces cannot remain within the final DNA molecule, demanding further action. Another enzyme, DNA polymerase I, steps in to remove all the temporary RNA primers. It then replaces these removed RNA nucleotides with the correct DNA nucleotides, filling the gaps. However, a tiny nick or gap still persists between the newly synthesized DNA fragments on the lagging strand. This final crucial sealing step requires a specific molecular glue to complete the seamless strand. DNA ligase, the ultimate molecular welder, arrives to join these fragments together perfectly. It forms strong phosphodiester bonds, finally creating a continuous, unbroken DNA strand. Simultaneously, an enzyme called topoisomerase relieves the intense coiling tension ahead of the moving fork. Without topoisomerase, the DNA helix would twist into an impossible, tangled knot, halting replication. This entire, incredibly complex molecular ballet occurs with astonishing speed and remarkable accuracy. The process includes built-in proofreading mechanisms, significantly reducing the occurrence of errors. DNA polymerase itself possesses an editing function, correcting most mismatched nucleotides immediately. Despite these safeguards, a tiny fraction of errors still manage to slip through the system. These rare, uncorrected changes in the genetic code are what scientists call mutations. Mutations, while sometimes detrimental, are the fundamental raw material driving all biological evolution. Understanding these intricate replication forks holds profound implications for human health and medicine. Defects in the replication machinery can directly lead to various serious human diseases. For example, uncontrolled cell division in cancer often stems from errors within DNA replication processes. Scientists are developing new drugs that specifically target these replication enzymes in cancer cells. Interfering with the replication fork can effectively halt tumor growth, offering innovative therapeutic strategies. Antiviral medications also frequently target viral replication forks, preventing infectious agents from multiplying. This microscopic cellular activity underpins heredity itself, passing traits reliably from parent to offspring. The elegance, precision, and efficiency of this biological copying system remain truly astounding to researchers. It's a testament to life's capacity for self-organization, even at the smallest, most fundamental levels. The DNA replication fork represents one of life's most sophisticated and essential molecular machines. If you found this fascinating, subscribe for more incredible stories from the hidden world within us. Share your thoughts in the comments below. What other microscopic marvels intrigue you? If you found this fascinating, subscribe for more incredible stories from the hidden world within us.